In this video, I will discuss how researchers can avoid bias by relating to the Dreu, Greer, Van Cleef, Shalvey and Handgraf 2011 research study, Experiment 1. The Dreu et al. 2011 is a study that investigated how oxytocin can increase in-group bias. One possible bias when conducting research is the researcher bias. Researcher bias is when the researchers may influence the research they conduct. This can, for example, occur during interviews if researchers knowingly or unknowingly are asking leading questions. Possible ways of reducing this is to use more than one researcher that can cross-check, analyze code, or verify the data that's produced. This is called researcher triangulation. In experimental research, the use of controlled variables are ways to reduce the influence of researcher bias and other confounding variables. Controlled variables are factors that are kept constant in an experiment in order not to bias the results. Controlled variables that may reduce researcher bias could be standardized instructions and a double-blind procedure. A double-blind procedure is when both the experimenter and participant are unaware of what experimental condition the participant is in. In the Dreu et al. 2011 experiment, the researchers excluded participants who were using drugs, were on medication, or had any medical or psychiatric illness. In addition, the experimenter was not present in the room when the participant completed the test. Other control measures were that the participants were randomly allocated to experimental conditions and that the test questions were presented to them in a random sequence. The researcher also used a single-blind procedure, which is when only participants are unaware of what experimental conditions they're in. This was to reduce participant bias, which is when participants may alter their behavior to conform to or react against the researcher's expectations. This may happen during interviews, as there is a tendency of social desirability, and participants may not be completely honest face-to-face. This can possibly be dealt with by asking open-ended questions without expecting particular answers and making sure participants are comfortable in sharing information. Some participants may also be more honest when answering questionnaires due to the anonymity. For experiments and naturalistic observations, participant bias can be reduced through deception or not informing the participants about the full aim of the study. This may be justified if the deception is not harmful, there's difficulty testing a hypothesis through other means, and participants are fully debriefed afterwards. The Dreu et al. 2011 deceived the participants as they were not informed about the true aim of the study. They unknowingly inhaled either a placebo or oxytocin, and the test also consisted of questions irrelevant to the research aim to mask what was actually measured, which was in-group bias. Finally, it's recommended that researchers attempt to reduce sampling bias, which is when the sample used in an experiment is not representative of its target population. The risk of sampling bias is reduced if the sample size is large enough. It is also recommended that the sample is homogeneous, meaning that participants are as similar to each other as possible. For small sample sizes, there is a risk for huge individual differences, which can lead to unreliable results. For experiments, the repeated measures design can be used to reduce the effect of individual differences. In this design, each participant takes part in all experimental conditions, so that ability will not be an issue. Such design also requires some randomization of the order of the experimental conditions to reduce the effect of order or practice. An effective way to reduce sampling bias is to use random sampling. This can be achieved by accessing records of the population and then choose and contact individuals randomly so that every individual in the population has the same chance of being chosen. The population can also be partitioned into subpopulations. The researcher will sample participants proportionally and randomly from each subpopulation. This method, which is called stratified sampling, deals with the problem of heterogeneous populations, as it divides the population into more homogeneous subpopulations that it samples randomly from. This would, for example, avoid the sampling of 9 boys and 1 girl from a population of 50 boys and 50 girls. In the Dreu et al. 2011 research study, 280 Dutch males were recruited through online volunteer sampling. As volunteer sampling is a non-random technique, there is a risk of a non-representative sample. However, the sample size was fairly large. To sum up, researcher, participant, and sampling biases can all be addressed through improved methodological procedures.